Okay, well, thank you very much. It's a true honor to be back and to see so many friendly faces. Many of you will not know that I've got some of my veterinary student classmates here in the room with us. I've mentors who are going to see their names and their pictures come up on the slides, and it's really great to see some of the faculty I've hired who are nearing retirement, and it's just really wonderful to be back and to accept this honor of the Schofield Lectureship. So I'm kind of old, and I don't think in the context of most of the people in the room. So I told Dr. Sharif that I could not give a current research lecture, seeing as I haven't done any research in 3 years. And he said, well, review your career. So I had to start way back at the beginning to get you in the right frame of mind so you know what the steps were as I went through the career. And you can decide if we did things right or did things wrong, but that's who I became, which is what this title means. So I started back in high school, and you'll see that I was interested in math and science. I was members of those clubs, none of this volleyball team or basketball team. No, we got to do thinking things. And I was the editor of the yearbook for 3 years and the proofreading editor. And so precision, I think, when I do testing of personality, I'm told I'm very precise. And I don't think of myself as that way, but that's what always come up. And I think being a proofreading editor sort of shows that. So you'll see I wanted to be a veterinarian for a long time. The alternate career, if I didn't get into OVC, was I was going to be a butcher. I really think I was interested in physics enough that I wanted to cut diamonds. I was prepared to go to Brussels and learn that trade. So I thought I should tell you what's the exciting science that I saw in high school that made me involved in this. And this is, see the slides aren't quite, they're all stretched. I'm sorry if they don't look quite right. It was polio vaccine. So I had classmates who wore braces who were post-polio. And as I was putting this together, I picked the one from Connaught Labs because U of T owned Connaught Labs. They were in vaccine development. The one in the upper corner is from Ottawa, 1,200 cases of polio in 1958 in Canada with 104 deaths, and it's gone up since last year. And so the Salk vaccine was huge, and my parents read a lot. We had all kinds of literature coming through the house. So I read all this stuff, and it was on the covers. It wasn't buried. This was the science, was polio, and then the Sabin vaccine. As kids, we paraded across the auditorium stage and got these vaccines. But there was something else going on, so I was very interested in these vaccines. And this was the pre-human space flights. So first dogs went up, and then non-human primates went up. And I was hugely interested in these animals in outer space. And I do have a pointer and the slides. This gentleman, and often in other publications, similar people, were identified as veterinarians. So I thought, this is really, truly wonderful, and I can be a veterinarian and do vaccine development, and that's my goal. So that was where I started from in the context of the science that was going on and how high profile that science was. So this is our entry-level class at OVC. This is the fellow I have married after I graduated, but only 2 of those women survived to graduate. Our class was 80, and you failed people out. So we failed about a quarter of the class in first year, and they brought it back up to 80 for second year, and we only graduated 63. So attrition was pretty high in those days. And we may look younger than you're expecting. We were the last kids to come in straight from high school. There was no pre-med, so we came directly into the veterinary program from high school. So the very first opportunity I had to go into my career was to get accepted at OVC. And as soon as I got here, I started collecting mentors. I've had many, many mentors, and I think it's really important for the graduate trainees and undergrads, if there are any in the audience, to know who are the mentors and how do you find them and what did they do for you. So the first was Dr. Bill Mitchell. He was our honorary class president, had been in the Air Force, had been president of the Canadian Vet Association, had been president of the Ontario Veterinary Association, And every time I had an issue, I ended up at his office, and he was always helpful, particularly at the end of first year when I decided to drop out, and I was going to wait a year and be in the inaugural class in marine biology. And he said, well, I have a friend out in Calgary. He might give you a job, and you could experience the practice, because I never had a pet. I was coming in from a science stream before I ever got to vet school. The second person was Dr. Amrit Singh, who some of you will know. He had just arrived from India. I think he taught us on the second or third day he was in Canada. And I had never looked down a microscope. I think everybody else in the class had, at least I felt they had. And they're busy looking at all these slides, and Anne can't focus on anything. He was very patient, telling me how to use a microscope. And then once I figured it out, or he got me figured out, he would pose these puzzles. He'd come over, there weren't very women in the class, and maybe nobody was really interested in looking at normal cells. But I found it fascinating, and he would create these little puzzles. Well, how are you going to tell that liver cell from this pancreatic duct cell? You know, how are you? And so I've used a microscope throughout my career, and I think it's all for the love of microscopes here. The next person I wanted to mention, I couldn't get a picture of. That was Mary Ray, who was an administrator in the dean's office. I was the student council president at my final year. It was really different in those days. Students couldn't have money. So Mary had to take the minutes of our meetings, and all the finances had to go through the dean's office. And so Mary and I got to be good friends. But she used to live next door to Schofield. And all the years I was on the faculty here, which was close to 20 years, Mary and I would come to the Schofield lectures. She was always call me when the Schofield lecture's on. We will go together. And she said, I don't understand anything the speakers say, but his ghost makes me go. So I think Schofield was a very, very interesting person. She lived just 2 doors down from the college there on Gordon Street. So this is what we looked like as vet students. This is my friend Marg, who was with me in first year, and me, and then the 2 women who were added at the beginning of second year, and we all graduated. And then, so how do you go on once you got your degree? How do you get into graduate work? How do you proceed to get this research stream? And Dr. Wilson, who's here, was a very kind faculty, new faculty member from the UK, and he encouraged me to apply for scholarship applications, and I happened to get a scholarship. And then my husband decided we ought to get married and go to Toronto and not do graduate work here in Guelph. And so Dr. Wilson, with other people, put together a set of interviews for me in Toronto, and I actually got an offer in all 8 labs, the immunology labs I looked at in Toronto. So I went off to U of T, and these are the people who I trained with. And I didn't really know until I was putting this together that these 2 men were both MDs. I knew that, but I didn't know neither had a PhD. And this was my PhD mentor. He was on a fellowship in Scotland and learned about this mouse. So he was trying to figure out what the thymus was doing in an immune context. We'd been taught here, it's big at birth, it regresses, it has no significance, you'll never need to know about the thymus. So I have learned about it since. He was doing surgical thymectomies on newborn mice, and that was how people studied the role of the thymus. And while he was overseas, he learned of this mouse that was found in Edinburgh. It's the nude mouse. It was the very first genetic immune deficiency. So you could breed these mice true, and there was no thymus. I looked histologically at every single one I used, and there was no thymus. So I was characterizing what's the immune deficit when there's no thymus. He was interested in transplantation antigens and went off to Africa for the last 1.5 years of my thesis, and this gentleman took over. Some of you will know Bun McCullough. He's considered the founder of stem cells. So he was a hematologist and did the first assays in the mouse for colony growth of stem cells, and then adapted those to culture. So he's been quite recognized with Jim Till, who was also at the Princess Margaret here in Toronto, where the old hospital. This is his picture when he was inducted into the Canadian Medical Hall of Fame. So I was mentored all along by people who were clinically active. This picture is the hospital. This was the front door. You'd go up the elevator, and this is a clinical ward. And I would walk through clinical wards, say hello to the nurses, be offered coffee or milk or anything. And the research lab was back here. And so it wasn't foreign to have mice and people mixed together in that hospital at that time. So what also accompanied me besides microscopy through my entire career was mouse genetics and the use of immune-deficient mice. And that led to doing foreign species grafting into those types of mice. And Dr. McCullough's issue was, doesn't matter, the projector can go out, you just take a piece of chalk, and if you can't explain your work to a lay audience with a piece of chalk, you're not yet ready to graduate. So clear communication was an essential thing for McCullough. Okay, so now I've got this graduate degree, and then what are you doing? How do you go forward? Well, my husband by that point had worked for a couple of practices, wanted to buy his own practice, and we bought a 40-year-old practice down in St. Catharines. That was the logo. So it had been a one-person practice for 40 years, didn't need 2 vets. So I kind of got a little bored just counting money. It isn't nearly as interesting as science, but I did all the business side of the practice, right? So I was reading the newspaper one night, and I read about the insert grants that had gone to Brock, and there were several people doing, I think, patch clamping and stuff that sort of vaguely sounded like I might fit in. So I went up to Brock, which was a relatively new university at that point, and the department head was very sympathetic. He said, we shouldn't have a PhD in this community. You can't find work, but we only have one person who works on mice. She's not here today. You got to come back. So that was Dr. Janet Rossant. And she was a world expert, or became one of the world experts in developmental biology, and a very significant mentor, number 5. So the pictures at the bottom are at the beginning of the anatomy lesson, right? I taught gross anatomy. This is a fertilized zygote, so you have the 2 pronuclei here. And this is almost any mammal, not quite everyone. The marsupials don't quite do it this way. But the cells divide. You get a 2-cell embryo, and then you come to a 4-cell. That's even got 3. Then you come to this stage. So what's interesting here is all of those cells are totipotent. You can break those embryos apart, stick them together in any combination, or just transfer them to a new pseudopregnant uterus, and anyone will give you a viable birth. But you look here, the same number of cells supposedly here in the morula is here, but there's a difference. And that difference is called compaction. So these cells, you'll notice they're not getting bigger, they're getting smaller each time. So that's the big cell. And this compact morula, something happens. And what happens is the first lineage differentiation of any cell in any mammal occurs. The result is a blastocyst, still free-floating, it's not attached to the mother at all. And the cells on the outside here are called trophoblast or trophectoderm, terrible word. They become the placenta. They can never give rise to the fetus that's born. And the inner cells are called the inner cell mass, and that gives you the fetus and can never become a placenta. So you go from cells that can give both to a commitment to a lineage, and 2 lineages develop. So that was what Janet studied, was that commitment to lineages. So just to make sure that you know that having an outstanding mentor is a really great thing, this is kind of a list of awards that Janet has held. And I starred this one. This is Canada's top international research award. 2015, she was the first woman to ever receive that. She chaired the working group for our funding agencies on stem cell that developed our national guidelines for stem cells. She was 10 years the chief of research at Sick Kids, and she's currently the president and scientific director of the Gairdner Foundation, which sort of pre-picks the Nobel laureates. So wonderful to have people like that writing references, but they also demand that you do science at the level that they think science should be done at. So they make you aware of that international expectation of excellence. So the question that I addressed in Janet's lab with her is a classic immune enigma. So if you take any gentleman in the room and any woman and you want to do a kidney transplant, you're going to go through all this matching. But they could have a child with no problem. And so how does the placenta, or the baby, conceptus, whatever you want to use, avoid the mother's immune system? The father's genetically different from the mother, and why does mammalian pregnancy work at all? So it's not a problem for mammals, but it's a good experimental question. So do immune cells either ignore, or are they suppressed? There's all kinds of literature on immune suppression in the uterus. Or do they actually respond and do something? Is it the placenta like a graft? Is it like a parasite? Is it like a tumor? What is this very strange structure that's allowed mammals to evolve? And what we did was ask this question: can these outside cells of that early blastocyst protect a foreign species? And this is the strategy, is micromanipulation, where you're doing surgery on those little tiny balls of cells. And the readout is this. This is a chimeric mouse, so it's a mouse made by fusing a black embryo with a white embryo. So it's two different animals, and that's what the word chimera means, has 4 parents. And this is probably the top experiment we did. At that point, we weren't into molecular biology yet. And so to place a mutation in a mouse on a specific chromosome, you did mapping. So you did breeding studies to mice that had a genetic marker where you knew where it was. And so we were trying to put genetic markers from this little wild mouse from Thailand into the standard lab mouse. And when you crossbred them, everybody died. And if you took those strange species, all the transfers are into the standard lab mouse, they all died. So embryo transfer didn't work, hybrids didn't work. And then Janet, with her micromanipulation skills, which she learned at Oxford and Cambridge before she married a Canadian and had to move to Hamilton, she put this inner cell mass of the one species in with the other, and she got viable offspring. If you did the reverse, so you had the identical embryo, but it put it in that cell envelope from the foreign species, they died. So then we made these aggregation chimeras in those very, very early stages. Those all lived. So some of the outside cells matched. And this experiment's never been repeated by anybody else. So she actually took a blastocyst, surgically cut the inner cell mass out, let it reseal, and then put the foreign inner cell mass in, and then she trusted me to do the embryo transfers afterwards, and those were all successful. And so what did that tell us? The conclusion was that for a viable birth, the uterus and the outside placental cells must match. And the embryo, the fetus, doesn't matter one iota. You can have a foreign species born if you've matched the placenta to the mother's uterus. So that's about where I was when I got a job. And that was 16 years after I got my vet degree. So this is not a fast track career. And I was hired here at OVC to teach gross anatomy of large animals. Well, you can see the expertise I had in that. Other than my undergrad degree, there was very limited expertise. The only horses I ever got close to were dead ones. I was terrified of horses. But I had a wonderful number of colleagues here, and I put Dr. Yamashiro and Dr. McInnes up. Dr. Yamashiro and I had started collaborating prior to me coming here. He was interested in any cell in the world that had a granule in it. He's an electron microscopist. He loved the granules. And the cells I was studying had granules. So we had actually met at a couple meetings, and he'd done a little bit of work with me. Dr. McInnes is one of the very many faculty who were delightful to teach with, as well as conduct research and discussions over a lot of wine. But every time I would have a harebrained scheme, like we're going to do PBL in first year and second year, and we'll move it from 2 weeks to 6 weeks, John would say, count me in, count me in, count me in. And so very many wonderful people here at the college. And then I also interacted with a lot of outstanding support staff, and particularly I've put Kim up, who remains a very good friend, and provided me with Mary Ray's house picture for this talk. So now we have to go to post-implantation anatomy in the mouse. So mouse pregnancy is about 3 weeks in length. It's not very long. And this is post-implantation by 2 days. And you've got a litter. This is just part of a uterine horn. And when you dissect this in half, this is all maternal tissue. In the mouse, the placenta is very invasive, and the mother makes a response called a decidua, and that's the same in humans. And so this white is all the mother's response. The red here is the embryonic crypt, so that's the old uterine lumen, and it's full of blood. And there's a little pale spot right there. That's all that is the conceptus tissue. This is 3 days later, so it's pretty rapid. There's your fetus. You can see the yolk sac. This is the nice placenta. There's the little bit of decidua. And when you cut across, you see that this is just a big sac of blood. So the placenta is the lung and heart and everything of the fetus. So what we were studying were these brown cells. So these are stained for lymphocytes. And we're up here in this decidua area. So one important point is these cells are never between the embryos. They're only right at the implantation site. They actually surround the blood vessels. And I put this word mesometrial here because these are called metrial gland cells. That's what they were called for years and years and years. And this is 2 days after implantation, so this stage. And this is before the placenta. And you see they're much more abundant. Here's our placenta, our umbilical cord, neural tube. And the cells aren't in the placenta itself, but they're right at the boundary in the maternal tissue. And this is the beginning of 3rd trimester, and you see the cells are gone. So they have an important role before the placenta circulation opens, and then they kind of do. So they're not around by term. And this is their name. And so our question was, what are these cells? We think they're lymphocytes. This is a lymphocyte stain. What's their function and what are they doing? And this is my next panel of colleagues. So you'll see these are pretty impressive guys. And they were all mouse breeders, and they were all really molecular biologists and would make wonderful mice. And then Anne would call and say, do you think I could have a fixed uterus? Would you send this from Europe to me? First one of these is Cox Tierhorst. Not only was he chief of immunology at Harvard, he was the editor of a number of journals, and he had the first mouse without an NK cell, a natural killer. So at the time we were working on these cells, they were defined as being a lymphocyte that was not a T cell and not a B cell. There were no specific markers for it. So these guys were the people who sorted all that out. His mouse was called TGE26, and I called Cox and got my nerve up and said, could you send me a uterus? He said, there's no point. There's no point. I've had the human pathologist and the veterinary pathologist at Harvard look at this mouse. It's fine. It's viable. The offspring are normal, and there's nothing interesting to find. I said, please will you do that or I'm leaving science. If I can't find something wrong, I am going to leave science. So he did. And it only has 1 to 2% of the cells that I've showed you in the stains. So these other people wrote papers with my students, provided mice. He's become interested himself, Mauro, in the mouse NK cell in the uterus. But Wayne and Jim both have trainees who have gone on to wonderful careers in reproductive immunology because of kind of interacting with me. Jim, I wanted to mention, his picture's bigger because I did the longest collaboration with him. And he had a mouse that was knocked out for the IL-2 receptor, for those that know some of this terminology. That receptor is required by NK cells to be viable, and his mouse had no NK cells. So this had 2 or 3%, this had none. And so I said, okay, now I've looked at the fixed uterus, would you please send me the breeding pairs? He said, no way. No way, I'm not sending you those mice. So, okay, this is all very interesting. He said, they get an autoimmune disease. They don't live beyond 4 months of age. And so I haven't published on this other mouse, but I've crossed it to a T and B cell deficient mouse, and I have a double knockout that's really nice. And I'll send you my unpublished mouse. So that's the kind of wonderful collaboration that you can't buy, it just happens. And so I think we published about 10 papers with him, and I used that mouse right to the end of my career. So this is 1990s, so about 30 years of using that. It was a beautiful mouse. But I would go to meetings of the Society for Natural Immunity. They met every 18 months. And that society was very small. It was only about 150 people would come. And I would be 1 or 2 or 3 of us doing reproduction. But these people who were the real leaders in the field were very receptive and very supportive and would say when they saw you, "What's new since we last saw you at the meeting?" And they would get you to get up and talk in the workshops and give your data. And so I found that that was very empowering. It made me read their papers, first off. But they got to talk to my students. They published papers with my student. If you said Wayne Yokoyama was on your paper, all of a sudden your paper got better reviews. They got published in better journals. And it really did promote my little small discipline within the immunology field very, very much. Okay, so I got these mice, and I got these implantation sites, and now I got to interpret what I'm seeing. So these two implantation sites, this is the normal one, and this is the mouse with no lymphocytes from Dr. DeSanto. The stain shows you there's lots of cells, there's no cells. And I ended up traipsing down to pathology at OVC on quite a regular basis. First with Dr. Dean Percy, who was a lab animal specialist and pathologist, and later to poor Dr. Foster, who was a placenta pathologist, but he did sheep. And so we would puzzle over these, what is going on in these implantation sites? And we published, our first paper about this was we had decidual cell immaturity, so the mother stromal cells didn't mature well. We had edema, that's what we interpreted. We'd stained for fat and we'd stained for protein. And I remember Dr. Foster saying, "Well, no tissue has air in it, Anne, so it's got to be edema." So we published that. And we had, as highlighted here, very unusual artery structure. And I remember arguing with Ruth Wilkie that these were veins and not arteries in this mouse. And eventually we found that all effects in this mouse with no lymphocytes could be reversed by injecting gamma interferon, which is a key product of NK cells. So we thought there was a lot of gene regulation in different tissues controlled by interferon, and that's what the cells were doing. Well, then I had my first postdoc who was from Japan, and I thought some of the trainees might like to know where trainees go after. He actually became the dean of 4 federated vet schools in Japan, but he was an electron microscopist, and he made these plastic casts by infusing plastic and letting it set in the arteries of the mother going into the placenta. So this is a typical normal mouse, and these are called spiral arteries. They're like a slinky toy, and you can see why they got their name. And those don't look right here. So the arteries are too narrow, and by not spiraling, we think they're too short. And so we concluded that there was a vascular effect where what happens in pregnancy to enhance the blood supply to this developing fetus, the blood has to be at a low blood pressure. And so the arteries get very flaccid and look like veins. And we thought that change was not occurring. And these were the studies that Dr. Sharif was referring to. 2 PhD students here, Jose Guimond, who went on, she's the senior grants coordinator at York University, and Ali Ashkar is a Tier 1 Canada Research Chair at McMaster. But they're still my most cited papers over my entire career, and they were done here. So the hypothesis that we came up with, again, came from a veterinary perspective rather than a human biology perspective. And we know that in litter-bearing animals, you could have 1 to, depends on the species, maybe 15 in the pig, fetuses. So you can't hardwire the mother's blood supply to the embryo, because each embryo has its own placenta. And so we said, okay, what cell moves around the body and can tell you where there's a metabolic change or whether there's a tissue antigen change is an immune cell. And so this is to represent an endothelial tip cell in the mother's blood supply. The tips are what grows, and we knew that these NK cells were making members of the angiokine family that makes blood vessels grow. VEGF is marked here. And we said, okay, you get the trophoblast from the embryo, the blood supply has to go there. And they're leading the way. They're paving a pathway for the blood vessels to follow into the implantation site. Well, now we come to more stories. I love telling stories. This is a woman who has she died this spring, had an honorary degree here from Guelph. And she was a reproductive immunologist. And I had been invited to speak in Rome. You don't get invited to Europe to speak that often as a symposium speaker. So I prepared, I thought, a really, really good talk. I dragged my husband. We saw Florence and Venice and all those places and end up in Rome. And I go to speak, and there's 8 people in the room. I'm going, well, this cost that meeting an awful lot of money to have me here. But this individual was standing at the back door. She wasn't in the room. She was talking to someone, and I didn't know who she was or anything about her. And shortly thereafter, she must have liked what she heard because I got invited to sit on an NIH panel in Washington. These are grants review panels, and we met 3 times a year and reviewed about 75 to 80 grants each time. And it was called Human Embryo and Development 1, HED1, and there was an HED2. So I was ad hocing on that panel for about 6 years. She actually ended up chairing the panel at one point. And then they decided non-US citizens could actually sit on the panels because they couldn't get enough reviewers. So I got a 4-year appointment. So I spent 10 years going to Washington multiple times to sit on these. Well, Joan was never satisfied with doing anything simple, and so she, with 3 other people, got a huge amount of money and developed a course that's still going called Frontiers in Reproduction. It's a 6-week course, hands-on wet labs. The faculty comes in for 1 or 2 days, runs a lab, and it's funded by the Rockefeller and the NIH and the Mellon Foundation. It's part of the US international programs, and it's still running. So I was involved in the first 6 years of that course and ran their immunology section and helped breeding and flushing all these embryos out of mice. Joan edited a lot of books, and she would invite chapters. She organized any number of conferences, and she always would invite me, or frequently invite me, to speak. She gave all kinds of advice very generously and freely. You didn't have to take it all, but it was given. And she would give grant writing advice. And I thought, that blurs. We became very good friends. But what were the benefits to me as a vet from Canada doing all this? Well, it totally changed my perspective in reproductive sciences. So this grant panel was looking at lung development, looking at brain development, looking at heart development. We would do small business grants where you're looking at monitors for babies, and then one clinician or pediatrician would say, well, that doesn't work because then they roll over, it'll dampen it, and this doesn't work. And you go, well, I wouldn't have ever thought of that. But it put my science much more into a human context. I don't think I was thinking human context at all. In fact, I didn't even really know what preeclampsia was because it doesn't occur in livestock. It only occurs in humans. And so leading on from these spiral arteries to preeclampsia and linking that histology to that disease came strictly out of this NIH experience. The ideas of others are being proposed for 5 to 7 years ahead. And so you would see where people thought a field was moving into areas you never expected. It's a lot of work on young women, sort of 11 to 13, having babies, and their cervix isn't long enough, and how are you going to, make sure these pregnancies go to term? It was, you never think of that in vet school. So I got many, many friends in disciplines I wasn't participating in, like I said, heart and lung, for example. And I was very frustrated about how hard and how long the hours were I was working. But I found many other people worked more than I did. They were directors of research institutes or editors of journals. And in addition to all the stuff I did, they did layers and layers more. So you couldn't really feel you were accomplishing anything very special. But I wanted to put this up because I think there are some grad students in, and this is a little discussion. These are just prompts to me. So Dr. Judy Head is an embryologist who taught at Southwestern in Dallas. And she worked on the NK cells of the rat uterus. I worked on the mouse uterus. Dr. Hunt worked in both species on macrophages. So we were all doing innate immunity in a rodent. And we were on this NIH panel together. And we had a young gentleman that came with me who was a postdoc, interested in that, but he was in a cancer lab. And we were giving him advice. And Judy said, what's the most marvelous thing of my whole career is teaching medical students. I adore the medical students, and teaching embryology to medical students is my love. She's retired, and she's still contract teaching embryology to medical students. And I said, what I like best is the graduate student teaching. I like taking this little rosebud and seeing it blossom into this independent, fully confident flower that's going off and going and doing its own career. Dr. Hunt says, well, that's all very good, but I want to influence national policy direction in research. And she went on to be the vice chancellor of her university, held $86 million in personal funding, linked 10 secondary institutions in the state of Canada and all their IT network, and she really was true to herself too. So I think you don't copy these people, and what I put in bold here are things I think I have repeated from knowing Joan can do it, then Anne can do it, and I'll come to those a little bit later. So just be true to yourself and follow what really motivates you, because nobody else can motivate you. Well, here at OVC you had to work on a livestock species, because at the time I was here, about 40% of my salary was paid by OMAFRA, and so I'm going, what am I going to work on? I mean, I'm a city kid. What am I going to work on? Well, pigs. I had this experience with Dr. Wilson, and so pigs weren't as threatening as anything else. And they have an epithelial chorial placenta, so I did learn that they're still teaching placenta biology here in the vet school. They don't in the medical school, but there's a range of 4 different levels of invasion of the baby into the mother. And it doesn't really happen in pigs, they just barely touch. So we could ask, we thought, do pigs have these NK cells? And do they go to implantation sites? And we could always only get cell mixtures because of the invasion in mouse. So we could say, well, if they don't attach, then we can get pure maternal cells and not worry about anything from the conceptus. And we also felt if we were looking at the 2 ends, because the human and mouse is one end and the pig the other, that if anything happened in both sets of studies, we could say, well, it's probably going to apply to all species in between. And then, luckily for us, there's 2 waves of fetal loss in pigs, and they've been very well defined in the animal science literature, one at day 20 and one at day 50 of 115-day gestation. So, okay, we're going to work on the pigs. So again, a career-making decision, and I had nearly 15 years of working on the pig, in addition to other things. So this brings up my next team of supporters and mentors. These are the faculty mentors. So Dr. Gordon King, who was in animal science, who had done ultrastructure of the implant sites of the pig. Dr. Better and Dr. Rayside, who's here, who were very influential. Ontario Pork funded a lot of this work in addition to OMAFRA. And Graham McRae, an industrial scientist down in Belleville at BioNiche, who we actually developed patents with for this work. This was my research team, my trainees, and I thought, again, their outcomes might be of interest. So Gord went to Scotland and did medicine, and just recently last year returned to Canada and is practicing in Toronto. Chandra was here at Guelph for a number of years running my research when I first went up to Guelph, or up to Queen's. And he's now the Associate Dean of Research and Grad Studies for the Faculty of Health Science at Queen's. And he works on endometriosis, so a human disease, but not pregnancy-related at all. And this was Colin Farrell. That was the English name he liked to use, but his real name was Yuan Fang. And Colin went on, and he's a professor in Shanghai. I visited his lab twice. And he's the director of Digestive Diseases Institute. So ask how all that happens. So what do mentors do? Well, I remember being invited to speak at an American Association of Animal Science meeting. The estimated audience was to be 800 people, so you got to practice to talk. And this whole crew here listened to this practice talk. And I kept saying, like I've been saying deliberately here, invasion. Placentas invade, and they jumped up and down, these 2 guys, and said, you cannot say that. You cannot. You will be a fool. No one will believe you. If you say that once in your talk, you will be out of the room. They just attach. They don't implant. But they also helped me write, and they were co-authors on grant applications. Poor Dr. Rayside here had every issue of the journal Biology of Reproduction that the library didn't have. So every time you wanted to see something, he was there. He nominated me to research societies, so you got discounts on going to meetings. They tutor trainees that in areas you have no expertise, like endocrinology. participate in journal clubs. We did so many journal clubs together. But really the message is they encourage, encourage, encourage. And I don't think you can have a really good research career without that encouragement from more senior people. Our pig work led us to change the main paradigm we were working on, that it wasn't the fetus or the placenta that was being rejected, that in fact the immune response was directed against the mother's blood vessels. Because in the pig we could show that all the changes in gene expression, we were using laser capture microdissection to take individual cells out of the tissue, all the changes occurred on the mother side before there was any change in the retarding fetus gene expression. So we think the baby just died because it lacked a blood supply. And that really focused the lab to blood vessels, which was something I knew nothing about. So I needed to learn about it, and this is that spiral artery remodeling changes to that affect 15% of human pregnancies. And the one we were interested in then was preeclampsia. And preeclampsia, the definition, it's a multisystemic disease. The main clinical presentation is hypertension in the last half of pregnancy. It's defined as being after week 20 of human gestation. Typically has nothing to do with nutrition, education, location in the world. And if it's not treated by delivering the conceptus, and the placenta is thought to be the cause of the disease, the women go on to convulsing, and they can have liver failure, blindness, and death. And so can the baby die. So it's kind of devastating if it's not managed. But under some conditions, such as artificial insemination, particularly with donor semen, so there hasn't been an intercourse exposure, it's up to 30% of those pregnancies are affected. So it's quite a concern within obstetric disease, never gets better. The only treatment still is to deliver the baby regardless of the gestational age of the baby. So we knew these NK cells made these blood vessel growing factors, and the one I was interested in was placenta growth factor. And this is data, and this is a pitch for biobanks. These data were not collected to have anything to do with looking at preeclampsia. They were a vitamin C trial in pregnancy. And so 600 some blood samples in a set of women that had been biobanked. But they went back and looked at this growth factor, and this is normal pregnancy in blue, so it goes up and then comes down, and this is birth. And these are all these angiokine disturbances are thought to be from products of the placenta. So the placenta is conceptus derived, it's not the mother, and so they look to be the biological marker from the placenta. The red line is women here who went on to preeclampsia. The black line is women who had overt preeclampsia at the time the blood sample was taken. So the key points here was there's a big gap in the levels, so there's a deficiency, but this deficiency could be traced really early in the first trimester. And so the NK cells in the human are most abundant, sort of in between the two red circles, because there is an equivalent population. So there was no model, no animal model for preeclampsia. It's one of the reasons I think a lot of progress hasn't been made. And so we thought, okay, we've got the mouse, we have no spiral artery remodeling, we have the first natural model for preeclampsia. And that's where Alistair Summerlee came in, another name who'd be familiar here. He came to Guelph to teach gross anatomy. I was on that higher-ed committee as well. And he was an expert for the purposes of this talk in carotid artery blood pressure recording. He did it in rats, and he said, okay, we can do a set of mice. And so these are terminal experiments. You make your measurement, and then the animal's killed. So we did this in a set of pregnant mice at different stages of pregnancy, and then I delivered this at an international meeting, this pilot data. And I never got to sit down. I walked back towards my seat, and each elbow was grabbed, and two men rushed me right out of the room. And they were people from this NIH panel. This was you don't work in the field, you know. And they said, you've got to stop that. You cannot do that research. Just stop. Don't do another single animal. Well, why won't I do another single animal? Because nobody will appreciate that work. You must do serial recording, just like the blood samples from the women. You must do serial samples on the same individual. So how do you take blood pressure in a mouse over the course of gestation? And the one guy was the director of the Heart Institute in Portland, so I decided I should pay attention. So this is the device you use to do blood pressure recording in rodents. They fit in this little holder, and here's the little keyhole, and that's where the sensor is there. And so this was great. Alistair and I took a whole year to fund the $11,000 for that equipment. And the experiment was going on beautifully. I had an MSc student doing that. We played music to relax the mice, and they were in dark black velvet surroundings. And then the mice actually went on to late pregnancy. We were interested, and they were too big for the holder. So we could not do the experiment. We never got the data recording where we were interested. Well, there's an alternative way of doing this, but it's very expensive. And that's chronic radiotelemetry, where you actually transplant a radio transmitter, and you have receivers, and you get all kinds of data continuously for 60 days. And it had never been reported in pregnant mice. Now, at that time, which was in the 80s, I was also trying to get human placenta to do some xenografting with. And HIV was a dastardly disease, absolutely fatal. And Guelph wouldn't release any placentas. So in 18 months, I got 6 placentas between 3 universities to do my studies. So this was all going on when I get this call from Queen's about, we have this research chair and we'd like you to come up. We think you'd be the right person. In my job interview, I met this gentleman, who later became my department head. And Mike was an expert in radiotelemetry in rats. And he said, I've got the surgeons in my lab. I got the platforms with the radio receiver, because each mouse has to be in a separate cage. He said, I think I've got 100 of them. And what else did we have? We have all the data analysis. I can teach your students how to do all this continuous recording, like millions of pieces of data. And he said, all you have to do for your CFI is get this little system in miniature, because the rat ones won't fit in the mouse. And they were worth $5,000, just that little bit of the system. The whole system's worth about $500,000. And so he said, I think 8 would be right. And so he calls down to St. Paul. And before I left his office, I had 15 minutes with him. I had the purchase order estimate price to put into my grant application. So it made it really easy to decide to go to Queen's, because I was going to answer this question and have this mouse model. So one of my students recorded, we did all the baseline. This is a summary as a delta from the mouse's baseline before it was mated, and that's what mouse blood pressure looks like. The 2 strains are different transplantation antigen backgrounds, so it looked to be universal in the pregnant mouse. And this all was so nice, because it fit the phases of our concept of mouse development. So before the embryo attaches, during attachment, that decidualization phase, then we get at this reversal down here, the opening of the placenta, a mature placenta, then we've just got a growth phase, and then we have postpartum, and that actually contradicted the existing literature done on tail cuff that they were hypertensive during delivery, but we had so much data you couldn't believe. And look where the NK cells are active in the uterus. So this is perfect. They do really have something to do with blood pressure control. So we did the experiment, and it didn't. This black line is our mouse with no lymphocytes, and it looks exactly the same as any other mouse. The statistical differences are for both strains compared to their own baseline, and we did it in first and second pregnancy, and it didn't make 1 iota. So we think all the obstetric textbooks are wrong, and the spiral artery remodeling has absolutely nothing to do with going hypertensive. So what are you going to do? What are you going to do? You've banked like decades on this hypothesis. Well, I was invited to the University of Tokyo by the president of the university as a distinguished visitor, come and do a sabbatical. So I was there for 2 months. And my mentor there said, well, you've got to talk to the veterinary students. I was in the vet faculty. And so I gave this, and afterwards Cuneo said, oh, Anne, they can't relate to you. They don't believe you. They don't know why you're here. Because these are the best students in all of Japan. These are the top. University of Tokyo is the best students, and we are the top elite stuff. And not one of these kids has ever failed at anything they ever attempted in their entire life. And here you've told them about a failure. And I said, oh my goodness. I didn't tell them about the other failure. So we had a mouse that had no placenta growth factor. The growth factor I was showing you was deficient. So we figured these mice would be really interesting. And we were going to do this radiotelemetry, and we couldn't even get the experiment started. So you clamp one carotid and put the telemeter in, and the other carotid's supposed to work, and you've got the circle of Willis around your pituitary, and everything's supposed to be fine. But these mice were having strokes, and they would never recover from the surgery. We couldn't even get them recovered, let alone get them bred. So we just couldn't do the experiment. So what do? You go away on another sabbatical. So I went down to Rochester, and I see Dr. Morant is here, and it was her colleagues down there and work with her that had me interested in blood vessels. And we adapted a technique called whole mount staining of live tissue to the mouse implantation site. So this is actually a genetic GFP embryo. This is the area my lymphocytes are in, and at high power, there's the lymphocyte stain. They're very abundant. And this little red stain coming across is the blood vessels that are here. And so we're sitting looking at these things. They have to do something. We know they're doing something with the blood vessels. Why are they there? What's going on? And then when we applied this to the mice with no lymphocytes, we got this. So you see the little round dots are the lymphocytes. There aren't any. Here we have nice branching blood vessels all stained. And here it's just all fuzzy, very fuzzy. But this is active angiogenesis, and this is a mature vessel. And what we felt the cells were doing then was aiding in the recruitment of the smooth muscle and the pruning of the vessels. And in fact, that made us reinterpret that edema that Dr. Foster and I had published on, that it wasn't edema, it was actual vessel immaturity that was giving us the big gaps in the tissue. And so I think the point of putting this in is it took many people, it took many years to come to a much more satisfactory explanation of what your system was telling you. Well, it's kind of interesting these animals stroked, because people who've had preeclampsia, their children are at fourfold higher risk of having a stroke. So we got really interested in what's placenta growth factor then doing in the brain, because the studies from the Helsinki cohort, which was thousands of people, said that there was no gain in coronary heart disease. And these data were collected in the 1930s and 40s, so these people are being studied at around 70 to 80 years of age. There's also about 50 papers in the literature on cognitive testing in children whose mother had preeclampsia, and they're thought to have a little bit lower IQ and some kinds of difference, and it's not due to the prematurity or anything else. It's related to the preeclampsia. So we asked the simple question, I thought at the time, do these mouse fetuses have abnormal vascular development in the brain? And if we don't use them as fetuses and have them as adults, can we do behavioral testing in the mice? Everybody else was, so why didn't we? And is there a cognitive impairment? So again, another team. And it's quite an interesting team. This is Dr. Sled from the Mouse Phenomics Center in Toronto, but a PhD student, MD PhD student, undergrad student, and 2 students from Brazil. And the first thing we did was map where the growth factor and its receptor are located in the mouse brain during development. So it was mapped in the adult, but it tends to be mapped to the forebrain rather than the middle brain or the hindbrain, and both the receptor and the factor are there. This is an antibody stain of these fetal brains, and you'll see it's around a nice blood vessel, but there's a lot more green than there is blood vessels. So the neurological tissue is also expressing that factor. So when we looked at the mice lacking it, we got into doing a number of fetal and neonatal mouse vascular assays that are standard assays in the vascular field. We found there were fewer of those tip cells. We got crossing over of veins that didn't happen in the retina. And we did a time course study here and showed that the vessels were not as abundant, and then the brain width started to lag behind. And here we were looking at the circle of Willis. And just to show you what that means, I think it's more dramatic. That's the difference. So we're not getting enough arborization of the blood vessels. This is the normal, and this is our mouse. So it looks like the vascular development of the brain requires this factor. So we moved on to a pilot study in children, and how did we justify this? So we had evidence that I haven't shown you that we had adult brain maldevelopment of the vessels. We also had reduced brain development in the adults. We had very specific behavioral deficits, and then I showed you the stroke data. So again, you use where you are to put together your experiments. And at Queen's, it was just a coincidence that I had everything I needed to move this work to children. So my colleague who'd helped in recruiting me was chair of obstetrics. He was following mothers who'd had preeclampsia for 5 years on their cardiovascular performance. And so we had the access to children who were 7 to 9 years old. Another person there, Dr. Reynolds, was the director of the NeuroDevNet, which was a National Centers of Excellence program in child brain development. There was a Canada research chair running a research-dedicated human MRI, and Kingston General Hospital was generous enough to give us $10,000 to do this. So we're going to undertake human research. First time in my career that I ever did human research directly. So these are the people. And again, to say the value of your students, your information on your career comes from everywhere. And this gentleman was in Germany when we contacted him. And this is my second year medical student. So I was doing small group learning. 3 medical students had to do a project related to research, and we met, for a week, once an hour for maybe 6 weeks. And Brandon said, well, I want to read this guy's paper because we have to do angiograms, and Queen's Research Center had never done an angiogram. And we had to have no contrast because these are children and we can't put anything in their brains. So we had to do the MRIs with no contrast medium. And he said, this guy's published on it, but it's going to cost me $35 to get the paper from the library. Have you got more access to the paper? And I said, no, but I could email him, send me the PDF. And he replied back from Hamburg in Germany and said, oh, that's really interesting what you're doing. I'm moving to Calgary. And he now holds a Canada Research Chair. But he's a computational expert who interpreted medical images. And so not only did the student link me through to Nels, he also volunteered to be the subject to set the parameters on the MRI. So this is my medical student circle of Willis in his brain. And I mean, this project wouldn't have gone ahead without that input. This is what we did to the children. It took a half day. And I'll point out to you that the parents consented, but I had to learn how you assent children. I even learned how to assent children who can't spell their name and write it on a form. We had 10 children, 5 girls and 5 boys, whose mothers had preeclampsia, and the matched controls came from this big maternal heart study. So 5 girls and 5 boys. So a total of 20 children in the study. They did 4 different protocols on the MRI machine. Then they did a thing called eye tracker movement that you can do on children as young as 1 year of age that does a lot of brain processing analysis. And then we did the full clinical cognitive testing. So it was a 4 to 5 hour protocol these kids would come in for. And they often came in on PD days and nights and Saturdays, and people were very happy to support your research by coming and working those extra days. Did we find anything? Yes, we think there's a unique preeclampsia offspring brain. This is the PEF1. The pale blue areas had larger volumes, so part of the brain stem was larger. And these dark blue areas, the blood vessels have a narrower radius. So that allowed us to say there was an impact, but it's a pilot study. And in fact, we need 300 children to do the final trial, and I wrote 11 grants and never funded them, so that's why you retire, right? The behavioral study contradicted the literature. It said lower IQ and mathematical difficulties. Our much more thorough investigation said they had visual spatial processing, which fits with the optic changes, the region that does optic processes changed. And memory recall, particularly for faces, and some of the psychological ones that involve faces, they were not very good at. This led to a lot of questions. Might they have more motor vehicle accidents? This eye tracker study is used for looking at fatigue in surgeons and truck drivers and that kind of thing. And so was there actually, because they couldn't position themselves in a context, either motor vehicle accidents or elevated risk of jobs? Were the strokes these people had more severe? Maybe they have the same frequency of strokes, but when they have a stroke, it's much more severe because there's not enough secondary vessels in the brain. And if you have a stroke, was recovery delayed? Did they not make as much progress in a recovery setting? I worked with a surgeon, or a neurosurgeon, and he had 100 people he questioned, and not one of them knew their mother's obstetric history. So it's not likely we're going to pursue that kind of line, but it says you really should ask your mother. Well, you can. So I really think that in academics, you really need to make sure others succeed you and are enthusiastic for the science, and you enable their success. And based on that Frontiers in Reproduction course, when I got to Queen's, we could get 20 or 30 placentas a week, so no 18 months to get 6. We could have all we wanted, and we developed a human placenta summer school where we brought teachers in from Europe and the States, and we trained 189 students from 39 countries in the 11 years that went on. It was a really, really successful program that hopefully is going to get resurrected because they've hired someone who's interested in placenta. I also had lots of time to do international teaching in my career. This is my collaborator, Dr. Yamada. This is a Queen's student. This is a Queen's student in his backyard in São Paulo State. And we had one of these exchanges programs for PhDs that we didn't think the mouse placenta was of economic development to Brazil and Canada, but we had to argue that. And one of the requirements in the grant was you had to have a joint publication. And we'd already published a dozen or so papers together, so we proposed a textbook. So this is an 800-page textbook we did where the grad students were actually the reviewers of all the chapters. And it was really a wonderful, fun project to do that took about a year and a half. I've also had lots of fun with lots of trainees. And we had a Christmas card contest at Queen's a couple of years ago, and we won for sugar and spice and everything, mice. So these are cage cards with, the barcode for your mouse number. And this is our GFP mouse here. We had lots and lots of funding all the way through my career, particularly from NSERC, but many, many various funding agencies, which also really makes your career a lot more fun than not having funding. And then I have a special thank you for my family. My parents all obviously read a lot of science, and that's how I got interested in science. My late husband, who died 2 years ago, my dogs, I've got 2 of this kind still, and both my children who chose not to go to university. This is my classmates. We were here for 50 years ago, and pointing out, don't count on the future, because we're down. About 1 in 5 of us didn't make the 50-year reunion, besides losing all those people in first and second year. And I still read the newspaper first thing in the morning. I got to have a cup of tea and read the newspaper. And I read this, and I was planning this talk, right? And I'm thinking, this is hilariously funny. Like, life just isn't like that. And so maybe it's more interesting to live your life and respond to what's going around, because, these failures get in the way, and that there's lots and lots of opportunities. And I think that Dr. Schofield, or perhaps his ghost, as Mary Ray said, and he's the 34th person in the National Cemetery in Korea, might agree. And while I was preparing the talk, I found this. So this was 2 years ago, and it's a museum exhibit in Seoul of the centenary of his first visit to Korea. So my closing remark is, what do you think arose in opportunities from his first decision to visit Korea? I don't think he thought a museum would celebrate it 100 years later, or you've had 50 years of Schofield lecturers come through here since that, and I really appreciate being recognized in his name. Well, thank you very much.
Dr. Croy, that was fantastic. Fantastic. Captivating, motivating, all of the above. Thank you very much. This is now time for me to invite Sonia Karim, President of Central Veterinary Student Association, to make a presentation.
Thank you so much for presenting today. We're really lucky to have individuals such as yourself work in our profession. So it's nothing but admirable. So the CVSA and OVC would like to present you with the Schofield Memorial Medal today.
Thank you so much.
We have Karen, our photographer. You know what, Sonia? Let me just go and grab Karen, and I'll be back in a minute.
I'm willing to take any questions if anybody has any questions.
Say we have a little bit of time for questions, maybe one or two burning questions, and Dr. Croy is going to be in OVC cafeteria for the next few minutes or so, and I'm sure that she would be more than happy to entertain any further questions. So, Karen, any questions?
John.
It was a lovely, lovely talk. Thanks so much for sharing that story. It's wonderful, wonderful. So why did you retire?
Why did I retire? Partly because the floor I was on in my research lab got turned into an animal colony, and I didn't have a lab or an office. That was an accelerant. My husband wasn't well, and he died 10 weeks after I retired, very suddenly. So I think the retirement I was planning, like, life got in the way, and it's not, you know. So I think it was the frustration of not getting the funding. And you get old, you get slower, you know. Yeah, I think a lot of the epigenetics and the molecular biology, you know, those kind of tools, they weren't the tools I wanted to embrace. I didn't really want to do DNA analysis and a lot of computer time. I really liked the hands-on biology. So I think a lot of that, you know, when I started at the Princess Margaret, it was like the war on cancer. And there was the initial tissue culture, like to be able to grow T and B cells and to get the urinary plaque assay to work in auger, like that's where I started. And so to move to, you know, epigenetics from that, you keep up for a certain year and then you think, oh, let the younger people do this. Yeah.
It's harder to learn new tricks.
Yeah.
Any
Okay.
more questions for Dr. Croy? So going once, twice. Please join us in the reception, for the reception in OVC cafeteria, and also please join me to thank very warmly Dr. Croy.
Thank you.